Welcome to a screencast of gene regulation. Over the next few clips, I'll be discussing a few ways to help you visualize concepts of gene regulation. Gene regulation is the cell's way of controlling gene expression. As you may be aware, organisms have thousands of genes that may be turned on or off at any time. We'll be looking at eukaryotes and prokaryotes. And one thing you need to be thinking about is why would organisms need to evolve pathways to turn on and off genes? To get started, we're going to be looking at eukaryotes. And some major players are the promoter regions, RNA polymerase, and some transcription factors. And there's two mechanisms that we'll be looking at, pre- and post-transcription regulation. The first is having to do with histone acetylation. Usually, the DNA will open up and will expose what's called a replication fork. And in this replication fork, it would allow either an RNA polymerase, or if there's replication occurring, a DNA polymerase to come in and actually either replicate, in the case of RNA polymerase, where you could actually have transcription occur. We have a histone protein. These histone proteins are what's allowing packaging to occur. So most of the time, your DNA is actually packaged. It's coiled up in these histone proteins, and it's tightly packed. And once it's packed, it packs up into several little coils, and altogether it's called a nucleosome. So RNA polymerase has an easier time accessing the DNA if that DNA isn't wound up into some histone protein. Alternatively, if it happens to be wound up, you can kind of see how difficult it would be for that RNA polymerase to kind of gain access. So here you have it wound up, and if it's wound up tightly, it's much more difficult for that RNA polymerase to find a foothold, in this case the promoter region, to actually bind and make an mRNA transcript. There's no binding occurring, and so in order for that to happen, you actually have to have what's called acetylation. And the acetylation basically kind of makes like bumps in such a way that it'll loosen the DNA from the histone protein, allowing there to be access. And if you have access, then that RNA polymerase can attach. Methylation of DNA is the addition of CH3 molecules to the DNA. And so as these methyl groups are added or attached to the DNA molecule, they change the shape of the molecule. As you can see here, it's almost like they add kinks to the DNA. And so when you have kinks in the DNA, almost inevitably you're going to have problems when you have RNA polymerase trying to attach. And so as you try to open up and find or make a replication fork, the replication fork as well is also filled with kinks. And so that RNA polymerase is going to have a much more difficult time finding an attachment point and actually transcribing the way that it should. So here you have a single strand of DNA showing methylation, and with the methylation, again, you have many kinks in the DNA, and if there are enough kinks, then an RNA polymerase cannot come in and bind like it normally would, and so therefore that methylated DNA is more likely to be turned off. And so what you just saw were some examples of pre-transcription regulation for eukaryotes. This example also has to do with eukaryotes, but in this case we have activator proteins that are in the process of enhancing transcription. So we have the folding over of this DNA molecule, and the pink little glob there are these activator proteins. And these activator proteins will allow a complex to basically generate along with RNA polymerase. And so when you have RNA polymerase present, that RNA polymerase will bind to this activator complex. And so this essentially is going to enhance transcription, almost kind of like an insurance that transcription is actually going to occur. Eukaryotes also have post-transcriptional regulation. 5' cap is available to allow the transcript to actually survive outside of the nucleus. Poly A tail is available to ensure that that transcript actually knows where it's going. And then you have splicing, which allows different combinations of genes to be put together through this process. Prokaryotic gene regulation is a little bit different. We have regulatory genes, operons, promoters, repressors, and co-repressors. And while the end result is the same in order to actually regulate gene expression, operons work a little bit differently. So when you hear the word operon, you should be thinking of the word operator. So operators are basically facilitators. They want to make sure that things are going according to plan. You also have regulatory genes, and these regulatory genes are present when there may need to be some sort of negative feedback. In the case of the picture shown above, you have the trip operon, and if there's too much tryptophan, that tryptophan is basically going to come back and shut down the production of those genes. In this case, we have a whole operon, which is represented by the straw, and there in the middle, you have this green little piece of clay, and that green little piece of clay is the operator. And then you have 3 genes that are following. We also have an RNA polymerase, which is represented by this yellow piece of clay. And then this small little piece of clay, this pink little piece of clay, is going to be our repressor protein. So we're looking at an example where we have a gene that is essentially turned on, and we want to get it to be turned off. So in order to do that, you have a regulatory gene that's going to code for a repressor. And that repressor itself is going to, at some point, bind to the operator, which is that green piece of clay. And in doing so, once it's bound, we'll have a change in the regulation of this gene. So genes X, Y, and Z are already turned on. We want to get this operon to somehow repress or turn off these genes. And so when you hear the word repress, it's just like as if this operon was going to say, no, we're not going to turn these genes on. We're not going to get any transcription. And in order for that to happen, we actually have to get a signal. And that signal comes in the form of a co-repressor, that little blue piece of clay. Once that co-repressor binds to the repressor protein, they create an enzyme complex. And as you guys already know, those enzyme complexes, once they form, you have a conformational change within those proteins. And the conformational change is such that when it finds that operator, it'll attach to the operator in a way that it'll kind of just stick. And once it sticks, okay, we'll basically have this obstruction. So RNA polymerase is looking for the promoter, and the promoter is basically calling it to it, say, hey, you need to attach here so that you can start transcription. But that obstruction is there. You have that repressor complex that is preventing that RNA polymerase from transcribing and making X, Y, and Z. Sometimes you have cases where you have a gene that's already turned off. So again, you have a promoter and operator region, but in this case, the operator region is bound with a repressor protein. And so we want to get that repressor protein to basically fall off. That active repressor, again, it acts as an obstruction, and so it prevents RNA polymerase from correctly binding and transcribing genes X, Y, and Z. In order to get that active repressor off that operator, we have to have an inducer. That inducer can bind to the active repressor, cause a conformational change that would allow it to basically kind of fall off the operator. RNA polymerase can then have access, bind to the promoter, and then go ahead and transcribe the genes that are in that DNA sequence. So why do organisms need these mechanisms? One reason is energetics. It would be energetically inefficient to produce enzymes or amino acids if that organism doesn't really need them. The other reason is for feedback mechanism. If you have too much of a good thing, you want to make sure that you don't overdo it. And so sometimes we need those signals to kind of shut down certain pathways, especially if those concentrations of amino acids or proteins are way too high. That concludes this presentation on gene regulation. I hope that these clips were able to help you out.